Hello and welcome back to episode 9 of the NCERT Biology 365 podcast. Today, we are finishing the organelle sections of Chapter 8 with centrosomes, centrioles, the nucleus, and microbodies. If you are following along in your textbook, we are on page 99. We'll break down the cartwheel architecture of the centrioles, clear up the exceptions of cells that live without a nucleus, and analyze chromosome structures. Let's get started. Let's start with the centrosome and its two perpendicular centrioles. Each centriole has an organization like a cartwheel. The outer ring is made of 9 evenly spaced peripheral fibrils of tubulin protein. But here is a key detail. Each peripheral fibril is a triplet, unlike the doublets we saw in cilia and flagella array. These centrioles form the basal bodies of cilia and flagella and organize the spindle apparatus in animal cells during division. Now let's look at the nucleus, first described by Robert Brown in 1831. Its internal chromatin was named by Fleming because it's stained with basic dyes. While we usually think of eukaryotic cells as having a nucleus, NCERT highlights two major exceptions that lack one: mature red blood cells in mammals and the sieve tube cells of vascular plants. Keep these two cell types in mind, as they are common choices in identification questions. Inside the nucleus, the nucleolus is a spherical structure that is not membrane-bound, meaning its contents are continuous with the surrounding nucleoplasm. This is the active site for ribosomal RNA, or rRNA, synthesis. Alongside it is the chromatin, composed of DNA, basic histone proteins, non-histone proteins, and RNA. NCERT provides a useful statistic here. A single human cell contains about 2 meters of DNA packed into 46 chromosomes. This packaging is essential for cell division. Chromosomes have a primary constriction called the centromere, with disc-shaped structures called kinetochores on its sides. Based on centromere position, we classify them into 4 types. Metacentric has a centromere in the middle. Submetacentric has it slightly off-center, creating a shorter P-arm and a longer Q-arm. Acrocentric has it close to the end, leaving one extremely short arm. Telocentric has a terminal centromere. Additionally, some chromosomes show a non-staining secondary constriction called a satellite. That concludes our look at centrosomes, centrioles, the nucleus, and microbodies, which also finishes our page-by-page study of Chapter 8. To test your memory on these final sections, click the link and take our Day 9 CBT practice quiz. Join our Telegram group to download today's handwritten notes sheet, and we will see you tomorrow for Day 10 as we run our final chapter review and full mock test.